Assalamu alaikum class. Hope you're all fine. Today we are going to study about innate immunity in detail. So this chapter is divided into 2 to 3 lectures, and this is the first part of this chapter. By the end of this lecture, you will be able to identify key features of innate immune system, how microbes are recognized by the immune system, what are the different receptors involved, and how specific this system is. Further, you'll be able to understand what are the different components of innate immunity and specifically the role of physical and chemical barriers in providing innate immune response to pathogens. Let's look at the general features of innate immunity. So innate immune response is the initial response or the first response to an invasion of a microbe, and it helps it to prevent, control, or eliminate it from the host. So it keeps the infection in check until the adaptive immune response is activated. The second feature of innate immunity is that it stimulates the adaptive immune response and can also influence the type or nature of adaptive response. We'll discuss this in adaptive immune responses in detail. And the third important feature is it not only recognizes the microbes and stimulate the adaptive response, it also is able to recognize the damaged or stressed cells. It eliminates it and also initiates tissue repair. So how this innate immune system recognizes the microbe? This is done by specific receptors which identify or recognize specific pattern on the microbes. These patterns are called pathogen associated molecular patterns or PAMPs. These patterns can be nucleic acids like RNA, single-stranded or double-stranded in case of viruses. They can be protein like pillin or flagellin in case of bacteria. They can be cell wall lipids like LPS or lipotecoic acid, like LPS in case of gram negative and lipotecoic acid in case of gram positive bacteria. Or they can also be carbohydrates like mannan, pectin, glucan in case of fungi. So these receptors are basically germline coded and they are present on different regions of the cells and recognize the PAMs when they are interact with them. So these receptors can be present on the plasma membrane as in case of TLR or toll-like receptors. And there are different types of TLR. Here in this case is TLR2 which recognizes the bacterial cell wall. So they can also be, these toll-like receptors can also be present on the surface of endosomes. Here in case they are TLR7, 8 and 9 which recognizes the viral genome by the DNA or RNA, and these can also be present in the cytoplasm like receptors called NLR or NOD-like receptors which recognize the bacterial cell wall lipid, or RLR which stands for RIG-like receptor which recognizes the viral RNA. Here you can also see another type of plasma membrane associated receptor called lectin-like receptor which recognizes the fungal polysaccharide. So as these receptors are germline coated, there are limited number of them and their diversity is low. But as they can recognize those patterns which are uh, common in most of the pathogens, so they can identify different microbes. Like here you can see a cell which have a receptor called Mannose receptor which is identical, but it can recognize different types of microbes. So the specificity of the innate response is due to the pathogen associated molecular patterns, and so that these immune cells can only recognize those microbes which have these PAMs on them by the help of these receptors. And uh, these receptors are able to differentiate between self and non-self. So they can only recognize those patterns which are present on the microbial surfaces, and in some cases they can also recognize the damaged cell as well. Now let's look at the tool-like receptors and how they bind to the pathogen in this video.
Pathogens such as bacteria, like this mean-looking fellow in gold, or a virus, represented as the spiky red particle, can penetrate initial defense barriers such as skin, which may be damaged. The bug or virus can then enter the bloodstream through intercellular traveling and evading innate effector cells in the skin layers. In the bloodstream, we see the virus encountering a major innate effector cell, the macrophage or dendritic cell. On the macrophage surface, TLRs are part of a series of receptors which may bind the viral particle. You can also see the virus entering the cell itself through phagocytosis or endocytosis, a merging of the viral envelope with the cell membrane. Endosomal TLRs recognize double or single-stranded nucleic acids which have been liberated from the virus through its uncoating of envelope and nucleocapsid protein layers in specific endosomes that fuse with the TLR endosomes. The signal that passes can be mediated through subsequent phosphorylation states of the chain of molecules in the pathway, demonstrated here by
The components of innate immunity is divided into two types, like first line of defense and second line of defense. The first line of defense of innate immune response consists of physical barriers, chemical and biochemical inhibitors or barriers, and normal flora. The second line of defense is composed of cellular barriers or cellular components, which are majorly composed of natural killer cell and phagocytic cells, soluble factors, and inflammatory barriers. We will discuss them one by one in detail. Physical barriers are part of first line of defense and include skin, mucosal membrane, lungs or respiratory tract, gastrointestinal tract, genitourinary tract, and eye. Skin is one of the most important physical barrier of a host. So the outermost layer of the skin is composed of a dead cell, which provides a physical barrier and a tough barrier for penetration of most of the bacteria, and also prevents the degradation caused by bacterial enzymes. So moreover, this epidermis layer is of dead cells is also periodically removed, which also removes the most of the bacteria or microbes present on the surface. So the skin is also provided with a specific type of lymphoid tissue called salt, which is skin associated lymphoid tissue. It prevents the access of the microbes to the bloodstream. Within these tissue, there are specific types of dendritic cells, which are resident dendritic cells. Therefore, they are called Langerhans cells in skin. These cells basically internalize the antigen and take them to the lymph nodes and activate T cells and B cell response. Infection can occur when the skin barrier is compromised or broken. A wound can serve as a point of entry for opportunistic pathogens, which can infect the skin tissue surrounding the wound and possibly spread to deeper tissue. Now let's look at the immunology of skin in detail with the help of
The skin is the body's primary barrier against physical insults and microbial pathogens. It represents a unique environment in which immune cells interact with skin cells to maintain tissue homeostasis and induce immune responses. The skin is composed of epidermis, dermis, and a subcutaneous fatty region. Commensal bacteria, fungi, and viruses living on the skin have beneficial effects in the protection against pathogens and in wound healing. The epidermis is composed of highly specialized epithelial cells known as keratinocytes. They are continuously replenished from just one layer of basal keratinocytes, which divide frequently. Dead cells called corneocytes form the outermost layer and are largely responsible for the barrier function of the skin. In the dermis, cells known as fibroblasts secrete elastin and collagen fibers that form a dense extracellular matrix. Blood capillaries irrigate the dermis, while lymph fluid is drained through lymphatic vessels to lymph nodes, specialized immune structures in which immune cells are activated after pathogen encounter. Diverse and functionally specialized immune cells populate the skin. In the epidermis, a specialized subset of dendritic cells, called Langerhans cells, sample antigen. They project dendrites upward towards the cornified epithelial layer and sample bacterial antigen such as toxins. Langerhans cells appear to be both anti-inflammatory and activatory depending on context. Dendritic cells in the dermis are highly efficient at capturing dead cells and presenting antigen, such as viruses, other intracellular pathogens, or skin-associated self-antigen, to T cells. If dendritic cells are the immune sentinels, T cells are the immune effectors. Healthy skin contains more than twice the number of T cells found in the blood.
A mucosal membrane lining the nose, mouth, lungs, urinary and digestive tract provide another non-specific physical barrier to potential pathogens. This mucosal membrane is composed of a layer of epithelial cells which are bounded by tight junctions, and these epithelial cells secrete a moist and sticky substance called mucus, which covers and protects the fragile layer of cell beneath it and traps debris and particulate matter, including microbes. This mucus also contains some secretions which are antimicrobial in nature, like lysozyme, which degrades the bacteria by degrading its cell wall. Lactoferrin is a protein which binds to the iron that is required by all pathogens. Because they are unable to compete for the free iron, the pathogens are deprived of their nutrients and die. And lactoperoxidases, which produces superoxide radicals, which are highly toxic to the microbes. So apart from these antimicrobial secretions, these mucosal membranes are also lined by mucosal associated lymphoid tissue like GARD and BARD, which we have discussed in our last lecture. So students, it's time for our first concept checkpoint. So the first question, you have to identify the PAMs in bacteria, viruses and fungi. You have to write one PAMs for each of them. Similarly, you have to identify one receptor present in the human host, or PRR, pathogen recognition receptors, which is specific to bacteria, one for virus and one for fungi. Right? Then in the second question, you have to write the different names of toll-like receptors, where each of them is located and what type of PAMs they recognize. Try to find out the answers and we will discuss them in our interactive session.